Voilà, donc là, ça va être en anglais aussi. So listen carefully. So Deka.
Thank you everyone for coming here tonight. My name is Victoria Ariel Rendón. I am the R&D liaison for Prometheus Life Technologies. Here at Prometheus, we enable at-scale commercial production of high-quality organoids in microgravity to improve biological testing and regenerative medicine. There are quite a few challenges in life science, but I'm going to focus on 3 of them, which are our target markets, which would be pharmaceuticals, research, and in the medical field, mainly regenerative medicine. Now, the issue of pharmaceuticals is that it is very, very expensive to develop a drug. One successful drug can cost up to $1.4 billion to develop, and around 90% of drug candidate trials fail in clinical trials. In research, around 100 million animals are sacrificed each year to develop the drugs, to develop research. And in regenerative medicine, around 106,000 people are on an organ transplant list waiting for a life-saving organ. And 17 people die on average each day waiting for these organs. Now, why are these issues present? Well, it's because in disease modeling and in drug development, our models are substantially based on animal models and on 2D cells. But there's issues with that. Animals are not humans. We can get close, but animal physiology is not the same as human physiology. Second, 2D cells do not represent us. And 3D, no? When we develop cells in a culture outside of the body, cells tend to develop flat and in 2D format because they cannot overcome the gravitational forces. In my body, we have natural matrices, and that's why my cells are able to develop into 3D, like you as well. And also, we lack regenerative medicine options in autologous tissues, meaning that for regenerative and personalized medicine, we need tissues that replicate the person and the patient in order to produce successful therapies. Now, the answer to this demand in the market are organoids. Organoids meet the demand for human tissue and biological models because they are more relevant to human needs. But there's quite a few issues with organoids. Organoids need 3D foreign material to grow because the cells cannot grow naturally without these artificial material. But the problem is with artificial material that could decrease the physiological relevance of our cells. If we want to replicate human conditions, we need to do it without artificial material. And not only that, because it's artificial material, it can increase batch-to-batch variability, especially among manufacturers. They can be smaller in size, and if a laboratory or company decides to do it themselves, they can be quite resource intensive. So our solution to these challenges and to the market need is our organoids produced in microgravity. Our organoids are developed in space. We can take advantage of the collapsing cost of accessing space. When we develop organoids in space, we grow them 100% naturally with absolutely no artificial material. And that is because in microgravity, the cells will aggregate together and form 3D structures, natural and more fidel to the human body. They are consistent in quality for reproducible results. They have a high degree of differentiation, meaning that they turn into the cells of the tissue type that we want. They are validated with tissue-specific markers, meaning that when we test them, compared to the Earth terrestrial models, they meet to the requirements that a regular terrestrial model needs to pass. They're consistent in size, and they are ready to use, meaning that a client can ask us for an organoid, and we will deliver. Oh, they took away that. No, I can't change that. Oh. Sorry. We have validated this our organoids on 2 proof of concept missions on the International Space Station, where we have developed nearly 500 organoids of 5 different tissue types with 100% yield, meaning that for every stem cell we send into space, we develop a full organoid. And this is a quick example. On the bottom is organoids developed in space. On top is our ground controlled. The bottom organoids are darker in color, signifying more functional tissue compared to the ground control that has more connective tissue and not the functional tissue that we need to test and develop disease and human models. Our technology and our proof of concept has been possible due to our unique team dynamic. Our team has exceptional experience in gravitational biology, in space, in business, and in biotechnology. And it is their expertise and knowledge that allows us to lead the market. So not only does Prometheus have the biotech technology, we also have the space knowledge. And now what Prometheus wants to do, we want to develop our R&D activities, and what better place to do it than here in the Grand Est region. Why? Ample opportunity for funding here. Great potential for collaborations and partnership, especially with prestigious and worldwide known institutions. There's exceptional infrastructure for startup development here. I think Alsace or Grand Est is #1 in the region. And we have the space ecosystem here, starting here at the International Space University. So here at Prometheus, we're hoping to utilize space for the benefit of all. Whoa!